It's a fun experiment. This is the one with all the bubbles in it.
Mhm.
So I tried to flame it.
All of it.
My dog just crop dusted me and I feel like I'm gonna barf.
Okay, so who am I missing? Am I missing anyone? Okay. We have all 12. Oh, we still have people joining in. Everybody in? Yes, no, maybe so. Missing one person.
Yeah, I'm coming.
Okay. Okay, I think everyone's on. Okay, let's get. Did I lose someone? Who did I lose? Who called themselves last place? Okay. Okay, let's get started. Even though I still feel like I'm missing someone. Who am I missing? I don't know. Ah, we're losing people. Okay. I'm gonna. Which of the following is not an enzyme in gene expression regulation? It is helicase.
Is there any way to make the screen bigger? This is my first time using one monitor. Is there any way to make the Kahoots? Oh, I figured it out. Never mind.
Okay. So guys, so helicase is the right answer to this. Helicase is an enzyme that is involved in unwinding the DNA when replication or something like that occurs, where you need to get in between the two strands, okay? Inducible enzymes, repressible enzymes, constitutive enzymes, they are all involved in some kind of regulation, right? They're inducing or repressing different genes and that expression itself. What do inducible enzymes do when specific substrate or nutrient is present? You guys are amazing. I love it. Okay, so inducible, induce. Okay, so everyone got that right. Wonderful. What do repressible enzymes do when a specific substrate or nutrient is present? Great job. Good. They do repress things and their expression of genes. Wonderful, wonderful. Constitutive enzymes are enzymes that are continually produced regardless of the chemical makeup of the environment. True or false? Okay, so it is true. So constitutive, think continuous. It's when so it continues doing what it does no matter what's going on in its environment, right? So even if some nutrient is not available or is available, it just keeps on doing what it's doing. So that's true. What type of control is involved when transcription occurs unless it is shut off by a regulator molecule? Ooh. Okay, almost even split, but more on the correct answer. Okay. So guys, negative control, when you think negative control, automatically think it's inhibiting. So that regulator molecule comes in and it's inhibiting it from moving further. Whereas positive control is transcription only starts if that regulator protein comes in and it's like, go on, dude, let's get some work going. Come on. You know, does that make sense? Okay. what type of control is involved when transcription occurs when a regulator molecule directly stimulates it. You guys are awesome. So positive control. What is a functioning unit of genomic DNA containing clusters of genes under the control of a single promoter?
I just read about that replication fork.
Okay, good. It is an operon. So remember, guys, with the structure of a gene. So I know it's a little confusing, but when you're involving multiple genes under one promoter, that's what's called an operon. You cannot just say that it is a promoter, okay? So I know a promoter is involved in every gene, but in some cases it could just be multiple genes can end up being controlled by a single promoter, and that entire unit that includes that promoter and the multiple genes, that's called an operon. So typically genes under an operon involves a function that kind of goes with one specific task, like it needs the multiple components to do that one task, right? Does that make sense? Are we okay? Okay. Which of the following is not a structural gene within the Lac operon? Good. So it is the repressor protein. That is not a structural gene, that is a regulator gene. So what do you guys know about these three genes within the Lac operon? They are structural genes, but what do they do? Any ideas?
What was the question again?
So what do these three genes do? So LAC-Z, LAC-Y, LAC-A.
Doesn't permease let it pass through, and the other one cuts it in half and separates the allolactose or whatever, the lactose. Yeah, and then the other one's not important.
Yes. So the Lac operon is involved in processing the sugar lactose, right? So in this case, LAC-Z produces an enzyme called beta-galactosidase. This guy comes in and cleaves what is called the beta-galactoside bond in lactose, which gives rise to glucose and galactose. There are some reactions that come in and kind of form allolactose, but that's a different story. Then we have LAC-Y. LAC-Y is the gene that gives rise to permease. Permease allows lactose to enter the cell. So it's helping it enter the cell. And then transacetylase, or LAC-A gene, that gives rise to transacetylase. This guy ends up detoxifying or removing any toxic compounds that were produced in the process. Does that make sense? Good. Yay. Okay. Attenuation involves the termination of mRNA synthesis. True or false? Yep, that is true. So attenuation is what is involved when it comes to terminating mRNA synthesis. So that is true. Attenuation of the trip operon functions by controlling what? Okay, who put translational termination when we're talking about mRNA synthesis? Where does the mRNA synthesis occur? Transcription, right? Chat box, what's up? It's okay. It happens. Don't do that on an exam though. But yes, it is during transcriptional termination. Oh my God, that is too much alliteration for me today. Okay, so it is during the termination stage of transcription where the trip operon ends up getting attenuated. Okay, so that's what's happening. This process occurs by controlling termination and transcription. Okay. Does that make sense though? Okay. Okay. Which term most appropriately refers to regulatory protein in prokaryotes? Good. Okay, come on, people. Out of all three of my sections, you guys are the only ones who chose translator. Like my last two sessions ended up not, like 0 people chose this.
I'm sorry, Nimi, my brain is going a little slow right now.
I understand. It's been a crazy week. But anyways, translator, that doesn't exist. At least not that I'm aware. Okay, so next ties one. RNA processing, guys, this is prokaryotes. We don't do RNA processing in prokaryotes to begin with. And that has nothing to do with regulatory protein. It's more, so RNA processing, remember guys, it's about the 5' capping, 3' polyadenylation, and then the splicing, right? All of this only occurs in eukaryotes. So no. And then helicase activation, that is in replication. It's just unwinding DNA. Oh. Okay, so DNA binding protein, great work. The bulk of DNA replication in E. coli is carried out by oh, okay. Everybody likes the first two polymerases, but not the third one. So DNA polymerase 1 and 3 are the ones that are more involved with this process, but the bulk of replication in E. coli is done by polymerase 3. Primase is just starting it off. It's literally priming that start. It's like putting a little marker, being like, hey, polymerase, come on, let's add on nucleotides here. That's what we need to do, you know? So that's why the answer with that is polymerase 3. To replicate a single-stranded circular DNA molecule, which enzyme is required besides DNA polymerase 3? Good. It is primase. So DNA polymerase 1, ligase, and helicase, those guys only come in when it's double-stranded, okay? So helicase, you're unwinding a double-stranded DNA. Whereas DNA polymerase 1, that comes in when it's on the lagging strand. It kind of goes through with this. So I have another figure in this thing where it shows it more clearly, where you have the different fragments going on, and they it's kind of stopping and then going to the next primer on that lagging strand. Does that somewhat make sense?
Yeah.
Okay, good, 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 good. Good, okay. Which enzyme is responsible for unzipping the DNA double helix? Good. That is helicase. Ligase is when it, so ligase ends up fixing any double-stranded breaks, any nicks in the DNA. It just comes in and it's like, let me repair you, and just joins them back together. Okay? Which enzyme can attach together strands of DNA without, well, with double strand breaks? Yes, it is ligase. We're getting people with helicase. I'm hoping that was an accident.
Don't worry, I got it right that time, Mimi.
Okay, good. But ligase is our answer with double-stranded DNA strand breaks, okay? Ooh, okay. Which is the DNA strand that the DNA polymerase constructs in the 5' to 3' direction? Continuously. Which I gave away the answer with that word. Good. It is the leading strand. Okay, so the lagging strand and the discontinuous strand, they are the same thing. It's because the whole, so it is discontinuous in the sense that it has to pace a little bit. It's like, okay, I need to make some here, then go back, make more, go back, another primer, and then kind of multiple primers, and then make it into little fragments, right? So that's how that ends up being little fragments, and that's how it ends up lagging behind this leading strand. So the leading strand is the one that happens continuously, whereas the lagging or the discontinuous strand is just saying that it's not continuous in the way that it adds on the nucleotides. Does that make sense?
Yeah.
Yes. I love it when people understand things. Okay. DNA polymerases add new nucleotides to which end of a growing strand? Ooh. Okay, who put N-terminus in DNA? N-terminus is where? Proteins, right? Amino acids. People, come on. Let's stick to the right molecule here. Okay, so it is the 3' end. So in this question, it's asking where is the next, so where is DNA polymerase adding to the nucleotide chain, right? So that goes on at the 3' end, right? You're adding the 5' end of the new nucleotide to the 3' end of that chain that is already being formed. Does that make sense?
Yeah, it does.
Wonderful, wonderful. My mouse does not like me today. Come here. Okay. The enzyme required only for replication of the ends of eukaryote chromosomes is wow. Now that is amazing. Okay, so telomeres are the ends of the eukaryotic chromosomes, and they get fixed up by telomerase. They make them look happy and pretty. So that's what telomerase does. Good work, good work. What is the central dogma of molecular biology? If anyone gets this wrong, I'm gonna cry. What? I'm happy that a
You went for the
majority of
meme?
you got it. What's happening?
DNA is everything.
Oh my gosh, I didn't realize that 3 people got it wrong. Oops.
Oh my god.
Nope. Nope. Mm-mm. Okay, so if you guys don't remember anything from genetics so far, remember this one, please. DNA to RNA to protein. I know DNA can be everything, but we need the RNA to translate everything, okay? Into proteins. We can't do anything without proteins. So, yeah, don't ever forget this. I've had upper level TAs come after me. Be like, what do you teach in genetics? You should be teaching them this. I'm like, I tell them. So don't forget it, okay? That's the answer. Okay, let's go on. Next one. RNAi attacks diseases caused by overexpression of a specific gene or normal expression of an abnormal gene product. True or false? Okay, so RNAi is a method in which they use to attack. So RNAi just is involved with interfering with the RNA that is produced, right? So if you want to get rid of an abnormal gene product, that is what you would do. You would use RNAi and it attacks the mRNA that comes out of it, and it's like degrading it all out. I'll put another video up for RNAi. That way you guys can have a more clear idea of what's going on. But that's essentially what RNAi ends up doing. DNA replication is semi-conservative, full conservative, none, both 1 and 2. Okay, so it is semi-conservative in the sense that if you see this guy, you have a new strain, you have the parent strain, right? Strand. I keep thinking strains. Okay, we have the parent strand and a new daughter strand coming in, right? So that's what it means when it's semi-conservative. When we say full conservative, it means that it's like one parent molecule, and then there's one new molecule that is just completely away from the parent molecule. So there would be one parent molecule and then new molecule, okay? So that's the differences between full conservative and semi-conservative. And this is what I meant earlier when I said, like, so a primer comes in here, it shows up there, and it goes from 5' to 3' in the opposite direction. Does that make sense? So primer here builds up, another primer builds up. It was a primer here, it built up here, and then the polymerase takes out the primers and ligase comes in, and it's like joining all of them together. So all of these little fragments are called Okazaki fragments. They get connected together and they're all happy, and they form that new molecule. Does that make sense? Okay, good. Yeah, one more question. What does DNA replication mean?
I think it's I don't know.
I don't know.
Give me the stub. Give me the stub. Mom, get the camera.
You do realize this is recorded, right?
Oh, I'm fine with that. I'm making learning fun.
That's my job. Anyways, who put this one? I'm curious now. Okay. It's okay. But yes, that is the answer. The process in which a copy of a DNA strand is formed. And that concludes our Kahoot session.
No.
Okay. Oop, okay. So let me just bring it back to this one. Yes. So that is pretty much I hope you guys enjoyed that.
I did. It
Thank
was my
you.
last spark of serotonin for the week.
Yeah.
Yeah, I thought it might be fun for the last day, you know.
Mimi, what are we gonna do without you? Plan a fail.
Nah, you guys will be fine. You just gotta figure out good ways to study.
Mimi, I'm gonna need you to teach me how to do Bio 330, please.
Bio, which one?
Bio 330 next semester.
330. Which one is that? Evolution? No.
Evolution and ecology.
Who's
Yeah, that is
teaching that?
not my strong suit. Nope. I've never taken that course, so that's one thing.
I almost had a
Who's
friend to
teaching
teach
that?
Charles Darwin.
Who's teaching
It's
that?
Hoff, Wang, and Hoff, it says.